So when we know how to produce RNA from DNA, now we have to learn how to use the produced RNA molecule. First, we have to recall the central dogma, stating that there are two steps leading from the gene to the protein. First, you have to perform transcription, which makes RNA molecule based on DNA template. And then you have to perform translation, which is transforming the sequence of RNA into the sequence of amino acids of the protein. The problem is that to produce a protein, you have to change the language in which the sequence is encoded. Because in RNA, you use the alphabet of 4 letters for nucleotides of RNA, and in proteins, you use the alphabet of 20 amino acids, let's say that. So it is impossible to just encode each amino acid by the single letter of bases in RNA. In order to do that, something evolved what is called the genetic code. And to stress, the genetic code is the way in which a sequence of bases in RNA encodes sequence of amino acids in the protein. I hope that from this moment on it will be clear that genetic code is not the sequence of the gene and not the sequence of mRNA, not any sequence at all. This is the way of encoding. If you see the number 20 for amino acids, it will be clear that we have to use the way of encoding which provides at least 20 different sequences. What happens in genetic code is encoding the single amino acid by the triplet of bases. 3 subsequent bases are called a codon, and such a codon has the special meaning and encodes 1 amino acid. But if you count all potential combinations of 3 using 4-letter alphabet of RNA, you will see that the whole number of different codons you can get is 64, much more than 20, which we really need for the amino acid. Due to that, some codons will encode the same amino acid. We call this property of genetic code redundancy, or we just say that genetic code is redundant. What does it mean? It means that some of the codons encode the same amino acid. Look. By the way, now we are checking how to use the table of the genetic code. If the unit of the code is a triplet of bases, you have to gradually find each of 3 bases to know which amino acid is encoded. So whatever is the form of the genetic code table, because they can look differently, the way of using them will be always the same. First, you have to find the part of the table which stands for the first base in the codon. So let's say that we are looking for the codon CUA. First, you have to find the part of the table which encodes for the C at the first position. Here, first position is the left margin of the table. The C is this blue row. So we know in which row we have to look. Now, if the second position in our codon is U, we have to find the column which stands for the second position U. So this is the first yellow column. Now we've got 4 codons in the column: CUU, CUC, CUA, and CUG. In this particular case, you don't have to look for any particular row because each codon here encodes the same amino acid, leucine. This is what we call redundancy. What is interesting is that however many codons may encode the same amino acid, but the same codon will never stand for different amino acids. CUU will always encode leucine, while CCU will always encode proline. This feature of genetic code, the very fact that in all organisms the same codon stands for the same amino acid, this property is called universality of the genetic code. We are going to discuss it a bit deeper in a moment. If you look at all the codons in the table, you will see that some of them are distinguished from the rest. There is a gray codon AUG. If you look into the key, you will see that it means initiation codon. In majority of cases, this is the first codon of each protein we can find. So, with exceptions, of course, because there are always some exceptions. But in general, all proteins will start with methionine amino acid encoded by the codon AUG. There are also 3 codons which encode for no amino acid at all. They are stop codons. Finding them in the sequence of mRNA means that here the whole protein production ends. The protein is ready. Later, we are going to see how it works. The sequence of amino acids in the protein is encoded by the gene. But what is important is remembering the central dogma, the very fact that to get a protein based on the gene, you have to perform the 2-step route. You cannot use the gene directly to produce a protein. First, you have to transcribe the gene into mRNA. Then you use mRNA to get the protein. So actually, talking about codons, you have to remember that it is not fully correct to refer to codons when reading the sequence of the gene. Look on the slide. On the right, you've got the sequence of the real gene. A, T, A, C, T, 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 and so on. This gene will in the future give you a protein, but not instantly. And when you look into the gene, you will find no codons. Nothing in nature can use DNA template to directly encode for the protein. But if you produce mRNA, then you can read the whole sequence, codon by codon, the triplet by triplet, and then you know which triplet stands for which amino acid. So I would advise you that actually using the term codon in your case should be restricted to describing mRNA and not the gene. If you remember splicing, it also explains that sometimes trying to look for codons in the gene sequence would be misleading. When you produce RNA directly in the process of transcription in the eukaryote, such raw mRNA cannot be used for protein production. First, it must be processed, including splicing. Splicing will remove fragments of mRNA. But when you look at the sequence of the gene, you don't know which sequences are introns to be removed and which are exons to stay and encode for amino acids. So this is another reason for not trying to look for codons in the sequence of the gene, right? Let's go further. However, I said that the genetic code is universal. It is, as usual in biology, full of exceptions. However, this exception is rare. So what we can say is that all organisms in the world basically use the set of 20 amino acids to build their proteins. And that this set of 20 amino acids is universally encoded by the same genetic code, what means that the same triplet of bases always stands for the same amino acid. But exceptions may happen in this form, that in this or that organism, the single amino acid in the set will be different. Quite often this amino acid is modified after protein production. And for instance, in some bacteria, cysteine will be not used, and instead of that you'll get selenecysteine. Why? Evolution. We are not going deeper into that. But of course, it doesn't mean that the same bacterium will use totally different set of amino acids for building its proteins than the rest of organisms. No, the basic set of 20 is the same. Only one amino acid is replaced by another one. And this is how these exceptions may look like. Particularly interesting are small exceptions in a set of proteins, in a set of amino acids encoded by mitochondrial genes. Later we are going to talk about it, but not today. Well, so let's look at our situation at this moment. We know that genetic code is universal in terms of how triplets of bases describe amino acids. In all organisms, with exceptions, the same triplet stands for the same amino acid. Another aspect of universality is that all organisms use the same set of 20 amino acids to build their proteins. Of course, it is not likely for all organisms in the world evolve separately exactly the same genetic code, exactly the same way of encoding amino acids, exactly the same set of amino acids to be encoded. As you probably remember, these 20 are not the only amino acids you can get, like ornithine, for instance, the amino acid which is produced, by the way, in the urea cycle in our cells, but ornithine is never used for building proteins. Similarly, with taurine, the amino acid you can find in energy drinks, like Red Bull, you don't use taurine for building proteins, however, it does exist. So what's the reason for all organisms in the world using only these 20, with exceptions, amino acids to build proteins? Probability tells us that evolving separately exactly the same genetic code is impossible. What is interpreted by scientists, like the evidence for all organisms in the world having the same ancestry. In other words, we had a common ancestor a long time ago, when life was only beginning. The same ancestor who had this genetic code, and for some reason this ancestor was so successful that now all organisms in the world are actually its descendants, inheriting, among other things, the same genetic code. Let's recall what are RNA types which we are going to use for the process of translation. First, the molecule you know the best, mRNA, messenger RNA, the molecule carrying the sequence of the future protein encoded. So, mRNA will stand as a template for the production of a protein. Now, tRNA, a molecule which carries amino acids and makes sure that each of these amino acids will find its proper place in the sequence of the protein. T stands for transfer RNA. The last RNA you have to know is rRNA, ribosomal RNA, which builds the whole ribosome with addition of some proteins. Why RNA? Why such a complex machinery is made of RNA instead of proteins? The answer should be known. If talking about nucleic acids, I ask you why something is like that, then probably this is about complementary base pairing. Using various molecules of RNA, we can use complementary base pairing as a way of joining them together. So it means that we already get quite a useful way of making sure that one molecule will bind to another properly and spontaneously, right? This is how it works. Of course, there are also other types of RNA, but we are not going to discuss them, focusing on these three because they participate in the process of translation. Let's look at mRNA first. Please do remember that IB is not going to ask you about the details of mRNA structure, except of the fact that it will have a cap at its 5' end and poly A tail at its 3' end, which both a cap and a poly A tail are the consequence of post-transcriptional processing, what you should know from the previous presentation. But functionally, not the whole mRNA molecule will be translated. So you have to know the term reading frame. The reading frame is this part of mRNA which will be directly translated. It starts with start codon and ends with stop codon. What is between the start codon and the stop codon is a reading frame, and this term must be something you memorize. The rest is not crucial. What is important is that mRNAs don't have any special shapes usually. About these cases when they can have some shape, we are going to talk later, but this is not crucial. Basically, what is important in mRNA structure is its sequence, but not its shape. So this is this rare case when we are focused directly on base by base sequence, not in the shape which molecule takes. The different situation is with tRNA. If you remember, this is this molecule which is going to carry an amino acid to the translation machinery and make sure that it goes to the proper place. The diagram you may most commonly find in various sources is like that. It's like clover leaf or something like that, and it stresses the fact that there are 3 loops within such a molecule created by self-hybridization of some fragments of mRNA with each other. So such pinheads are formed, and each tRNA molecule will have always such 3 loops, and this is why we stress this shape in this form. But in reality, the molecule is not flat. These pinhead loops are folded to form 3-dimensional structure. So this shape drawn here is only sort of a simplified version. But it stresses 2 crucial parts of tRNA molecule. Each of them will have CCA sequence at 3' end. And this 3' end is where the amino acid will be bound by the covalent bond. It is important because covalent bond, as you remember, is quite strong. So this is a safety-proof mechanism making sure that amino acid will not get lost and will not detach until this is the good moment. Another important part of each tRNA molecule is anticodon. Anticodon is at the opposite end of at the opposite part of tRNA molecule than CCA end. And anticodon should bring to your memory the term codon, of course. So if there are some codons, then anticodon probably must have something to do with them, and it does. Actually, anticodon is complementary to the codon. Of course, there are many different codons to be used, right? Like 64 minus stop codons which don't have their tRNAs. And it means that for each codon that we can potentially find on mRNA, there will be proper anticodon on some tRNA molecule. So if there is a codon encoding for, let's say, methionine, the methionine carrying tRNA must have a complementary anticodon on its loop to find the proper codon, right? This is how amino acids find their places within the protein. Well, there is a proper codon. tRNA comes with a proper anticodon, bringing proper amino acid. So the real decoder of RNA sequence is tRNA molecule. tRNA is capable of recognizing the codon and positioning the proper amino acid where it is needed. But of course, we are going to use 20 different amino acids. Each of them will be bound to 1 or more special codons. For each of potential codons, a separate tRNA molecule must exist with its own separate anticodon. So if all codons are 64 minus 3, it gives us 61. We need 61 tRNA molecules with different anticodons, and each of these tRNAs will carry its own amino acid. So tRNA is actually not a one molecule, but the whole family of very similar molecules differing only in the anticodon loop and the amino acid they are carrying. Getting the idea? If not, try to listen once more to this fragment or go to our webpage and watch some other videos linked there to clarify the issue. We are going to move forward. Because I said that each tRNA will have a different anticodon, and due to that, it will carry a proper amino acid. So how do they know? How do tRNA molecules know which amino acid they should carry? And the answer is a proper enzyme. For your knowledge, the name is tRNA activating enzyme. However, its real name is much more complex. It is amino acyl tRNA synthetase, but you don't have to memorize it. Let's focus on tRNA activating enzyme as the idea. Its work is to combine proper tRNA with a proper anticodon to the relevant amino acid. And this combination will be performed using the ester bond, which is quite strong. So some energy will be used for this process in the form of ATP molecule. So if we've got the enzyme, you of course have to recall what you know about how enzymes work. Of course, they have active sites, and these active site shapes are specific for particular substrates. In this case, particular substrates are the amino acid, tRNA molecule, and ATP molecule. I will go back here to the animation once more to let you look at the whole process happening on the right panel step by step. Please try to focus and look. First, the amino acid arrives to the active site and binds. Then ATP molecule arrives, binds, and hydrolysis provides energy for the change of the active site. Then tRNA may bind, and due to removal of the remains of ATP, the active site changes its shape slightly to form the bond between tRNA and amino acid. Then the molecule is released. Which aspects are important here? Of course, what is crucial is that this is how the enzyme makes sure that the proper amino acid is bound to the proper tRNA. Of course, it means that if this action must be specific, then there will be the whole family of tRNA activating enzymes, similarly to the whole family of tRNA molecules. Each tRNA with each amino acid will have its own activating enzyme, which is specific enough to recognize this particular amino acid. And this particular anticodon of tRNA molecule, not to confuse them with other tRNA molecules or other amino acids. So this is a good reference to the idea of enzyme substrate specificity, right? You can use this example to illustrate how specific this whole reaction is, taking into account that each tRNA differs from other tRNAs only by the sequence of 3 bases of anticodon. So the specificity of the active site in the enzyme must be high enough to distinguish this particular needed tRNA from the rest of tRNAs that could appear in the proximity of the enzyme. Even if there is a whole bunch of different tRNAs, the enzyme will recognize only the proper one because it is specific for its own anticodon it should recognize. The second thing which you should take into account is, of course, energy use in this reaction. ATP is needed to provide energy for the formation of ester bond between the amino acid and tRNA, or actually it provides energy for the enzyme to change the shape sufficiently to make the bond form, right? This case, when the ATP arrives and then phosphate groups are separated from ATP, and this is what provides energy for the reaction. This is the example of substrate level phosphorylation. If you don't remember what is substrate level phosphorylation, go back to the part of our cell energetics unit when we talked about ATP and its roles in the organism. Then you can recall what phosphorylation is, namely substrate level phosphorylation. The third issue to focus on here is the induced fit model. This is not only the fact that amino acid and tRNA are recognized by the enzyme due its specificity, but also the fact that to catalyze the reaction between amino acid and tRNA enzyme, the active site of the enzyme must change its shape. So this is exactly what induced fit model states. This is not like after binding to the active site, some magical catalysis occurs. This catalysis is the fact that the enzyme changes shape, bringing two substrates closer to each other in the way which is sufficient for the bond to form. Induced fit model is something you should know, and if you need any example to illustrate the idea of induced fit model, you can use the case of tRNA activating enzyme and how it behaves when performing its job. Well, the last player in the game, the ribosome. It has a small subunit and a large subunit. Both are made mostly of RNA, however, with protein addition. Each of subunits has a different role to play. A small subunit will have mRNA binding sites. So this is a subunit which is responsible for recognizing mRNA and finding the place where it should start. On the contrary to that, large subunit has nothing specific to do with mRNA, but it has three tRNA binding sites, E, P, and A sites, and they will help to arrange tRNAs carrying amino acids in the way which will provide proper sequence of amino acids. So, in P site, you will always have tRNA holding the growing polypeptide. This is why a P site is actually abbreviation for peptidyloside. In A site, you will always find a tRNA carrying the new individual amino acid. This is why A stands for amino acid site. And finally, E is exit site, the site to locate temporarily the empty tRNA without amino acid to get rid of it. So, to stress separate functions of small and large subunit. Small subunit is focused on mRNA, finding the proper place to start, while large subunit is focused on arranging tRNAs in the proper positions. In addition to that, large subunit contains so-called catalytic part, which will form peptide bonds between amino acids. We are going to see it later. You have to remember that the shape of a ribosome is not as neat as this diagram suggests. In reality, a ribosome is a rather large and quite complex structure composed not only of RNA but also of proteins, and it makes it really hard to visualize how it looks like and at the same time to show how different parts interplay. So, depending on the function Diagrams and models of ribosomes may have a different complexity. These models I wanted to show here are mostly to stress this complexity. But the models which are used in diagrams of translation are much simpler because their function is helping you in understanding how it works, and complexity would not help. So on diagrams, we are going to use this nice bubble-like model of a ribosome and to stress some additional things. The ribosome is not only going to bind mRNA but also move along it, translating the sequence of codons step by step, codon by codon. What is important is that it starts on 5' end of mRNA and moves towards 3' end. What is different than it was for processes involving polymerases on DNA, because polymerases move along their template strands from 3 to 5, but a ribosome moves along RNA template from 5 to 3' end. Why it is important, you will see later. What also is made visible by this diagram is that the real role of large subunit is to let two tRNA molecules position their amino acids in such a proximity that the catalytic part of a ribosome can create a peptide bond between subsequent amino acids. But this diagram also illustrates one more important thing. The formation of a peptide bond is not the only thing which happens when two tRNAs position amino acids close to each other. You can see that when the bond is formed, another thing happens, and the amino acid which was first bound to the tRNA on P side will be cut off from its tRNA, so the whole growing polypeptide is transferred onto tRNA which was in A side. This is why the process of building the polypeptide is not only elongation of the molecule, but also translocation of the growing molecule with each step onto a different tRNA. It seems a bit strange, but it has its logical explanation. Later we are going to see. So the whole process of translation consists actually of three separate phases. The first is initiation, and it lasts till both subunits of a ribosome join together on mRNA and find the proper position to start the next phase. So initiation ends when the whole translation complex is assembled and ready to work. Then there is elongation translocation. Elongation because amino acids will be joined together and the polypeptide will grow and get longer. Translocation because growing polypeptide chain will be translocated onto the next and the next and the next tRNA. Elongation translocation will be repeated as many times as needed to translate the whole length of reading frame on mRNA, right? So the more codons, the more repeats of elongation translocation sequences. And this phase ends when the stop codon finds its place in A site. Then we've got termination. The protein is released and the whole translation complex is dismantled. Initiation is slightly different in prokaryotes and eukaryotes, but the basic rule is quite similar. So, the first thing is that when the initiation process starts, small subunit is the most important player because it is expected to find mRNA and its proper part to start. So, small ribosomal subunit will find the 5' end of mRNA, bind there, and move along the mRNA molecule till it finds the start codon. As you can see, on the right diagram, so eukaryotic initiation. The small subunit is already carrying the first tRNA with the first amino acid with it. So, due to the fact that we said that in majority of cases the first codon is AUG, the first tRNA already bound to small subunit has codon UAC. This is anti-codon to AUG, and it carries methionine amino acid. And the small subunit moves along mRNA to find the stored codon. It clicks when the anticodon of tRNA finds the codon of AUG, the first codon of reading frame. So when they find each other, tRNA binds with its anticodon to the codon of mRNA by, of course, complementary base pairing. And this is how the first amino acid of the protein, methionine, is positioned properly to signalize the beginning of a translation. Then, when methionine on tRNA is at the spot, the large subunit knows where to sit. It must sit in such a way that tRNA with methionine will come into the P site. So this is why tRNA with the first amino acid is needed, to let the large subunit know where to sit. And in this way, it establishes the normal situation of the ribosome performing translation. P position is taken by tRNA carrying whatever is the polypeptide at this moment. Exit site is ready to accept the free tRNA, and A site is ready to accept the tRNA bringing the new amino acid. After such a situation happens, so small subunit, large subunit, and methionine carrying tRNA are at their positions, we may say that initiation step is finished and the next part of translation may go on. The next part is elongation translocation. Elongation translocation process consists of the same sequence of steps repeated many times. So, we start with a situation when tRNA with the first amino acid is in P site of a ribosome, as at the upper panel. Then A site exposes the next codon. The first codon, AUG, is in P site and holds tRNA for methionine. But the next codon is now exposed in A site. Each of these sites covers one codon only. So, when the next codon is exposed by being placed in A site, this is a signal for proper tRNA to arrive and bring the new amino acid. Which amino acid should arrive? Such an amino acid which is encoded by the codon in A site. How does amino acid know that this is the position for this particular amino acid? Because its tRNA has the complementary anticodon. So, however, there are many tRNAs grouped around the translation complex, only this one will bind within A site, which has proper complementary anticodon to the codon exposed. The role of P and A sites is to provide proper space for tRNAs. But who performs the real reading of codons are tRNAs on their own. They recognize codons by their anticodons and bind where they are expected. Okay, so let's say that the tRNA, proper tRNA, arrives with its amino acid and sits in A site. Then you get the situation which is presented on the second part of the diagram. In P site there is tRNA with an amino acid, and in A site is another tRNA with another amino acid. So now catalytic part of the ribosome will form the peptide bond between these two amino acids, creating a dipeptide. I will stress it once more because these are reactions you know already. Formation of peptide bond. You know it. You drew the diagram. I will recall the diagram later. Catalytic part of the ribosome forms the peptide bond between two amino acids, and now we've got a dipeptide. This was elongation step. Elongation because two separate amino acids are now bound together to form a dipeptide, which is of course longer than an individual amino acid. But this is not the only thing which happens here. Together with formation of a peptide bond, the first tRNA is removed from its amino acid, or actually the amino acid is removed from tRNA. Now the whole dipeptide is bound to tRNA, which is in A side, and in P side the empty tRNA is left. This process, translocation of the dipeptide onto the tRNA in A site, causes the movement of the whole ribosome one codon forward instantly after translocation. So when ribosome cuts amino acid from tRNA in P site and locates it on the tRNA in A site, then the whole ribosome moves forward from 5' to 3', one codon forward, what leads to the situation shown on the bottom panel. Now, the tRNA holding the dipeptide is in P site. tRNA without any amino acid goes to the exit site and is instantly expelled from the ribosome. And in A site, there is a next codon exposed, and the new tRNA may arrive bringing the new amino acid here. And the whole sequence of events will repeat. So if new tRNA arrives to the A site, it will bring a new amino acid. Then the ribosome will cut the dipeptide from the tRNA in P site, move it onto the tRNA in A site and form a peptide bond, so tripeptide now will be present, and the whole ribosome will move one step forward. And the whole sequence will be repeated as many times as needed to get the whole protein. If you get the idea, that's great. If you don't yet, go to the web page and watch animations linked there to clarify the situation. The ribosome moves forward as long as it may find in A site the codon encoding the next tRNA. But finally, the moment will come when in A site there is codon not encoding for any amino acid. Instead, this is stop codon. There is no tRNA to bind there. Instead of that, there is a special protein called release factor which will bind to this site. What may be interesting for you is that this protein has the shape very similar to the tRNA, so this is how it finds its way. tRNA would be better. So if there is any relevant tRNA with the special anticodon, it will bind faster. But if there is no relevant tRNA and the A site remains empty for some time, because what is in A site is the stop codon which has no tRNA, then being empty for some time, A site allows for binding of release factor, and it means the end of translation, termination of translation. Binding of release factor will lead to the separation of polypeptide from its tRNA. Polypeptide is let free, tRNA is let go, and at this moment the whole ribosome disassembles. The end of the translation of this particular protein. This is a good moment to stress the fact that each component participating in translation can be recycled. The same mRNA can be read many times. The ribosome however it splits at the end of translation, may reassemble once more and perform another translation. The fact that tRNAs are recycled is known from the very beginning, so after being used to bring a proper amino acid, the empty tRNA may fall out of the ribosome and be charged once more by tRNA activating enzyme with a new amino acid, and may be used many times even within the same translation, not talking about other translations. And of course, the release factor is also recyclable. So this is quite efficient machinery enabling performation of the same translation and producing many identical proteins very quickly. Here, I would like to stress once more that the reaction catalyzed by the ribosome is the reaction you know already, the reaction of peptide bond formation. So, of course, IB may recall this fact, and even if they are mostly focused at the moment in extracting from you what you know about the translation process, they may also ask you about the very chemical reaction of formation of a peptide bond. So this is what you should be able to draw with addition of water molecule, which is also formed. The first part of the drawing are two amino acids separate from each other, and the good practice is giving each of these amino acids different side chains to stress the fact that these two amino acids can be different. Then you show that after the reaction the peptide bond is formed between the carboxyl group of one amino acid and amino group of another amino acid, and you should also draw the fact that water molecule is formed in this process. If you don't remember it fully, go to the presentation about proteins and check how it should look like. Now, to complete our knowledge about translation, we have to say some words about where it happens. So the natural site for translation is a cytoplasm. Of course, in prokaryotes there is no other possibility. But in eukaryotes, it is also the original site of translation. So, RNA is produced in the nucleus, then it is processed, then it goes to the cytoplasm, and then it may be used for translation if the protein is supposed to stay in the cell. But if the cell wants to produce a protein to be secreted from the cell, then the ribosome will bind to the RAF endoplasmic reticulum, and then, during being produced, the protein will be stuffed into the reticulum and then sent to the Golgi apparatus and then out of the cell. So once more, the proteins to stay in the cell are translated in cytoplasm, while proteins to go out of the cell will be produced on RAF endoplasmic reticulum. But this is the same process of translation, and the ribosome is the same. And the only difference is that to produce the protein for export, the ribosome will sit on RAF endoplasmic reticulum. If you don't remember how it looks like, go to our favorite movie about the inner life of the cell. This difference is neatly presented there, and you can see the similarities and differences of the translation, the cell protein and the export protein. This diagram will bring out two additional issues to take into account. Of course, it stresses the fact that while in prokaryotes there is no other way than to instantly produce a protein in the cytoplasm, in eukaryotes it is necessary to transport RNA from the nucleus first to make it ready for translation. And as you maybe remember, between the moment when RNA is produced by transcription and the moment when it is used in translation, some things must happen. This is RNA processing, including formation of a cap and a poly A tail, and splicing, and only after that the mature RNA will be transported to the cytoplasm and used. So, actually, in eukaryotic cells, transcription and translation are separated not only by the site but also by the time. It takes some time to process RNA and transport it to the cytoplasm. On the contrary to that, as you can see on the left panel, in prokaryotes, there is no need to separate these two processes in time. So quite often it does not happen. The RNA is still being produced while its 5' end is already exposed to ribosomes, and they may start reading sequence of mRNA and translating. This is what is shown on the diagram. Such a situation explains why ribosomes start their work on 5' end. As you remember, polymerases, including RNA polymerase performing transcription, move along the DNA template from 3' to 5' end, and they produce their mRNA strand from 5' end to 3' end. What means that 5' end goes out from the template first, and it is already exposed to ribosome, while the opposite end may be still in production. Such a situation can be quite often seen on electron micrographs of prokaryotes. You can see that one end of mRNA is not ready yet, while the opposite end is already used by ribosomes and translated. And another thing which is stressed by these diagrams, and maybe you already asked this question to yourselves, that on these RNAs there is not one ribosome performing translation, but the whole set of them. This is quite a common situation. Such a structure, many ribosomes translating the same molecule of RNA at the same time, is called a polysome. A polysome enables production of the same protein in large amounts very quickly. It would take much more time if the next translation had to wait till the first one is completed, but there is no such a need. So if only the first ribosome moves forward enough for another ribosome to sit at its back, the next ribosome may start the next translation. It is very easy. And quite often these structures may attract the attention of people looking at the electron micrographs. As you can see on the right panel, this is a very typical distinct look of such a polysome. Moreover, you can see easily which end is a 5' end and which end of RNA is a 3' end, because we know that ribosomes start at 5' end. You can distinguish which ribosome is more advanced in the translation because it will carry the longer protein. Can you see that? So look at the middle right panel. At 5' ends, there are ribosomes just starting translation. You can see individual ribosomes, but you don't see their proteins yet because these proteins are very short at this moment. But the more forward is the ribosome, the more advanced it is in producing a protein, so the protein is longer and longer. The longer is the protein you can see, the farther in the translation is the ribosome. So you can see the 5' end as this one full of ribosomes without visible proteins, just starting, and 3' end of mRNA as the end full of ribosomes carrying long, almost ready proteins. Such a task may be posed to you. In addition to recognizing a polysome on the photo, you may be asked to decide about which end is 5', which end is 3', due to your knowledge about the length of the protein at the beginning and at the end of translation. You should be also able to say in which direction ribosomes are moving. What else can be seen on these micrographs is a difference between the size of a ribosome, the protein, and mRNA. Look at this middle photo. You cannot see mRNA at all. It is just too thin to be visualized on the photo with this particular quality. Right. Ribosomes are the biggest here because they are huge balls of RNA and protein, and they are not only well visible but also quite dark. And growing proteins, however rather small, are better visible than RNA because a protein has bigger thickness than an RNA strand. The top photo has better resolution, so even if a difference in sizes is still visible, you can see the strand of mRNA stretched between individual ribosomes. However, it is visible that mRNA is much thinner than the growing protein, right? Look at the left part of this slide. This is a diagram showing the specific situation that may take place in eukaryotic cells. If you remember, post-transcriptional processing of RNA includes adding poly A tail at 3' end of mRNA and a cap at 5' end. It may serve this function as you can see here. It helps to clip the beginning with the end of mRNA, closing the whole molecule into a circle. Why is it important? Because when at the end of translation, so arriving to 3' end of a translated mRNA, the ribosome separates in two subunits, and it is supposed to be recycled and used in another translation. But if you want to produce the same protein in many examples, then we want to start the new translation of the same molecule as quickly as possible. Joining the 3' with 5' end makes them closer to each other, and then it increases probability that right after completing one translation, subunits of a ribosome will find the beginning of the same mRNA and will join together to start a new translation. So instead of linear mRNA molecule, such a circular polysome makes recycling of ribosomes much faster and the process of producing many copies of the same protein more efficient. Now, there is time to recall the central dogma, the golden rule of molecular genetics. We know already that genes encode proteins. We know already that the coding of genes must be always done in two steps. First, the gene must be transcribed to form RNA. Then RNA is translated to form proteins. And for a long time it was thought to be the general rule that one gene stands for one protein. Now you have to know at least 3 exceptions from this rule, and you know them already. However, I will list and explain them for you to make sure that you get them. So, the original idea was one gene, one polypeptide. Now, exceptions you know. Some genes will be not translated at all. These are genes encoding for tRNAs and rRNAs. The RNAs have different functions than being translated. So the function of the gene is only to encode for tRNA or rRNA and not to be translated later, right? So the first exception is that genes for tRNA and rRNA are not translated at all, and they give no polypeptides because this is not their function. The second exception you know is alternative splicing. The gene is transcribed and it gives raw mRNA, but then in different cells, RNA may be processed differently by splicing, different set of introns will be removed, and finally you get different RNAs starting from the same gene. One gene, but many different RNAs after alternative splicing, and each RNA gives a different polypeptide. One gene, different processed RNAs, and different proteins at the end. The third exception is that there are operon genes, polycistronic genes they are called, when one promoter starting one gene encodes instantly for some different tRNAs. In this case, one gene instantly gives you different RNAs and later different proteins. Operons like lac operon are examples of this exception. And of course you also know already the fourth exception. These are cases where the mature protein must be composed of many subunits. So proteins having the quaternary level of structure are encoded by many genes. Each gene encodes for one RNA, and each RNA gives a different subunit of the future protein. But the protein will work only if these different subunits are arranged together. 4 differences, at least 3 of them should be used if you are asked to discuss the idea that one gene encodes one polypeptide. Now, this is a good moment to go to the presentation about proteins and recall everything what you know already about the protein structure. Go to the presentation and read and listen once more to what are primary, secondary, tertiary, quaternary structures, which bonds are used to stabilize them, what meaning has each level of this structure. Only after recalling this information, you can say that you know everything what you should know about translation and its product, a protein. Thank you.